Welcome. My name is Rob McLeod from the University of Utah and the Scientific Computing and Imaging Institute. This is another in our series of documentation videos showing how you can actually use the UncertainSCI toolkit to solve and enhance some of your modeling and simulation projects. In this case, specifically in the area of bioelectricity in the heart.
Okay, as Rob mentioned, we're going to talk. Jake and I are going to talk about some specific bioelectricity cases in the cardiac domain, and just to give you an idea of how we've used UncertainSCI to set up uncertainty quantification pipelines and applications. And of course, later there'll be some neuromodulation examples by Suminsha Ren%. So we talked a little bit about this pipeline before. We're starting with, when we do UQ, we start with a forward modeling pipeline in most cases where we have our input parameters, our model, and then the solutions. And then the model, for most applications, we treat, at least with you from the UncertainSCI standpoint, we treat it as a black box. And then, but what we don't know, if we have some sort of uncertainty of the input parameters, what the uncertainty on the output parameters are. So that's where we bring UncertainSCI into this, where we, do some kind of quantification of the input distributions, send those to UncertainSCI, where we generate samples and design a parameterization scheme, sampling scheme, and then run the model for that parameter set, and then build the polynomial chaos emulator, and then calculate the output distributions. So, even when you have a tool like UncertainSCI, which is non-invasive and fairly easy to use, at least we think, there are certain challenges to performing uncertainty quantification, knowing the input uncertainty and analyzing and visualizing the results. These are still challenges even for us who are, actively working in this on the software and in this domain. And I'm sure you all agree that these are challenging and they're active areas of research. People dedicate large amounts of times on how to do both quantify input uncertainty as well as visualize and analyze results. These are both active areas of research, and unfortunately, the approach to both of these things, in most cases, is a combination of intuition, literature, and data. And hopefully, it's more on the latter end of the list, towards data and literature than towards intuition. But sometimes you have to start somewhere. So we're going to talk about two case studies today. One is segmentation shape variability in ECGI and the uncertainty generated from that. And the other one is the uncertainty of biodomain conductivities in the static biodomain simulation. So first up, we're going to talk about segmentation shape uncertainty quantification in ECGI. And this is based off the presentation I just did in FIMH, which I misspelled, unfortunately. But I did a poster presentation about this in the conference that just wrapped up, and that's available. Of course, the paper is available online. So the uncertainty quantification, basically this problem started out as part of this the Consortium for ECG Imaging. We have a bunch of work groups, and one of them is the modeling error work group. We wanted to basically understand where, when you do ECGI or electrocardiographic imaging, you have to make a model, and that model could have errors, and we want to understand the effect of those errors on the ECGI. And so what we started with was segmentation. So we had a bunch of users in the CEI group segment this particular patient, and we saw some significant variability just looking at the data. We wanted to quantify those the effects of those error, both in terms of actual differences in the segmentation as well, and also in the effects on ECGI. So we did a preliminary study a couple years ago where we took those segmentations, we performed ECGI on each of those segmentations, and we computed, just based on those 9 samples, or I think in this case it was 6 samples, the variance generated from just having multiple different geometries instead of just the assumed one ground truth geometry. And we found some significant variation when we just used different geometries. And so what we wanted to do is, build on that kind of preliminary finding and understand more, analytically and quantitatively the impact of that segmentation variability on ECGI pipelines. And as I mentioned, the segmentations were done by the members of the CI working group, and it's done on patient data that is found on the Edgar database, which is available online. But what we did was we took this patient, we got 9 segmentations from people in the Consortium for ECG Imaging, and in order to quantify the variation, we used a statistical shape modeling tool called ShapeWorks. ShapeWorks is a correspondence-based shape analysis tool where we take the input segmentations and it finds correspondences on all the different samples within the cohort. And then from those correspondence sets, it generates shape statistics. And with those shape statistics, we can also generate what we call a shape model, a parameterized shape model. And so ShapeWorks is a useful tool. It was a useful tool in this case for quantifying the uncertainty of the geometry, in this case, of the segmentation geometry. And as we'll look a little bit we'll take a little bit of a look at the ShapeWorks outputs, the shape model. Here are the correspondence sets, and the colors kind of indicate correspondences across the different geometries. And it's obviously there are some duplicate colors, but basically the correspondences will match colors. But if you look at, so this is all the correspondences, and from these correspondences we made what we call a shape space from the principal component analysis of these correspondence sets. And what we get is basically 4 modes of variation in this case, which encompasses about 90% of the variability, the total variability. And as you can see, as this is an animation, hopefully it's actually updating fast enough for you all to see. This is an animation kind of progressing through each of the modes of variation along the shape axis, and how the shape model varies with each of these principal modes of variation. And so basically from this, this is the basis of our shape model. We have these axes. We know basically how each of our segmentation samples in our cohort, how they project onto each of these axes of variation, these shape modes. And then from that, we can generate distributions, statistics along each of these axes, and then use those statistics to drive our statistical shape model in our uncertainty quantification pipeline. So as a quick recap, from ShapeWorks, we get our shape model, which includes the principal component axes, the shape modes, and then distribution along each of these axes, which we basically, due to the limited sample size, we just set to uniform, but it also gave us kind of the standard deviations, and basically the range of variation along each of these axes, which we were able to use in our uncertainty quantification pipeline. So here's kind of the overall pipeline. We talked about this first part, the statistical shape modeling tool, shape model, where we took the segmentations, used ShapeWorks to generate the shape model, which is the axes of variations and the distributions. We fed those into UncertainSCI to generate our samples, and use those samples in our ECGI pipeline to generate the uncertainty quantification results, which we'll talk a little bit more about. So I'm not going to talk too much about some of the specific implementation of the pipeline because we want to leave time for Jake to talk about his example. But I do want to talk a little bit about the the analyzing and visualization of the results, because this is the second problem that is challenging that hopefully will help you all get some ideas on ways that you could approach your uncertainty quantification problems and how you can analyze or visualize your results. So in our case, the output of our ECGI UQ pipeline were potentials that are 3D and temporal signals. So very challenging to visualize even without the uncertainty distributions. And we also have local activation times, which are nonlinearly responsive to the change in potential. So the way that we kind of broke this down is somewhat typical when you have higher dimensional data, such as 3D or temporal data, is you try to reduce the dimensionality as much as possible so that you could use 1 and 2D visualization techniques. So first off, we took the temporal signal and we computed the RMS for over the epicardial surface to kind of show how the variation changed in time. And we also looked at individual tracings, which I unfortunately did not get into, which I don't have in these slides. But basically, we can see from these plots how the general epicardial potential changes over time, over the cardiac cycle. And as you might expect, and what we're seeing here is the median RMS potential over the epicardial surface, as well as the quantile bands in the red, and of course the darker bands being the closer to the median. So as we can see, the variation or the uncertainty is proportional to the amplitude of the signal, which I don't think is surprising to anyone. One of the interesting things about one of the things we can look at with UncertainSCI is also the sensitivity. And the nice thing about sensitivity is it's normalized to amplitude. So it kind of gives us an idea of, if we look at it in the temporal domain, and this is the global sensitivity, as we talked about before. The global sensitivity is all the interactions and how it contributes to the variance. So we can see over time how each of the parameters interact with the total the epicardial potential RMS signal. So the interesting thing about the sensitivity here is basically it's showing that what we know from our shape model, which is designed to pull out the most. So ShapeWorks is designed to extract the most significant modes of variation using and that's the principal component analysis. And so what we're seeing in the sensitivity and the sensitivity metrics is basically that we've managed to do that. The first 3 or 4 modes of variation or axes of variation contribute most to the uncertainty of the epicardial potential, at least in the RMS. And then the interactions, it shows that since the interactive states are relatively low amplitude, that they contribute very little to the overall uncertainty, so that the individual modes of variation are mostly independent in terms of how they interact with the uncertainty space. So, I mean, this is not necessarily, this is just sometimes a sanity check for people who are running their own uncertainty quantification pipelines. And sometimes that's helpful, even if you don't necessarily put them in your final paper, which this didn't actually go in my FMIH paper. So looking at the spatial distribution of the uncertainty was a bit of a challenge. There's some approaches which show a bunch of different spatial maps of the different statistics, like the mean, the standard deviation, and maybe the sensitivities, and those can be effective for sure. They can be more precise. We came up with this method of using cylinders assigned to the surface to indicate some metric of uncertainty. In this case, the standard deviation, or it could be the variance or the quantile ranges, or whatever value you choose. And the advantage of this is that it condenses the information into one image that you can look and get an idea of how the variation and also the mean kind of it's easier to overlap those two sets of data. However, the challenge with this is that I haven't found a good way to precisely indicate the values of that are represented by these cylinders. As you see here, I have an arrow pointed to this specific cylinder indicating that particular standard deviation, but it's not the most precise way of visualizing scientific data. So, I mean, it has its benefits and drawbacks. We can see this middle figure compared to, say, this right figure, which is the standard deviation averaged over the temporally averaged over the cardiac cycle, and we see similar areas of high variation, but this is more precise because we can tell exactly the variation in this region is 1.1 millivolts, and everywhere else is close to 0. But it's also when you have to look at two figures, it's got its drawbacks as well. One of the interesting things about this particular example was, when I first presented the concept of the shape variability impacting the epicardial potential solutions from ECGI, was how much that actually mattered to clinical metrics such as local activation times. And I didn't have a good answer at first, but UncertainSCI actually helped find an answer for that was somewhat surprising to me, although in retrospect, it kind of makes sense. So when we looked at the spatial distribution of activation times, we found regions of really high variation. So for instance, in these areas, you get a standard deviation as high as 67 milliseconds, which is a fairly significant amount of the total QRS duration. But we also, interestingly, have areas of very low variation, almost 0, in terms of standard deviation. And so this was really helpful to visualize spatially, because what we see is, this is the activation map. We see that, in areas with high gradient in the activation times, that's where we get the high variation, and that helped us correlate to, and that helps us understand how this variation and the actual signal are related, or the local activation time signal are related. And the other thing that I did in this particular visualization that I found helpful was to plot the quantiles instead of, say, for instance, the mean plus standard deviation, to show, kind of, this would be like the max and min of a sample set, but because it's generated through a PCE, there's not really a max and min, so that the quantile extremes are a similar type of, or an analogous way to represent, analogous to the max and min. So there was a lot of questions as to why there's so much variability, and in order to do that, we looked at a bunch of signals, and this is also, sometimes this brute force approach is helpful. Because this particular plot, I think, was helpful for me, because when I looked at these data, this is, of course, just to explain this a little bit, this is the most variable locations on the epicardial surface in terms of the local activation times, and we plotted the epicardial signal, the epicardial potential, and the derivative, the temporal derivative, in red. So the blue is the epicardial signal, the red is the temporal derivative, and the black lines are the computed activation times, local activation times. And we see that in these highly variable locations on the epicardial surface, that even like the change in the epicardial potential could be significant due to this segmentation variability. But even in cases where it's not that significant, say for instance in lead 121, this doesn't change that much, but it changes just enough to change which peak the algorithm, which in this case was the minimum dV/dt temporal derivative, chose for the local activation time, and basically from an early activation site to a late activation site, which explains the high variability that we see in the spatial domain. So, I mean, this just provides some. This is just a case study to provide some examples for you. Hopefully, it's helpful for you to see some examples of how we visualize uncertain quantification. We certainly love to hear your ideas. If there's definitely new strategies emerging all the time, and we don't have all the answers, and some of you probably have better answers than I do or than we do on how to visualize these things. And so we look forward to seeing yours. But next up, we're actually going to have Jake talk about his case study, conductivity concerning quantification in static bidomain.
Okay, and so like Jess said, I'm just going to be presenting another cardiac bioelectricity case study. And whereas Jess focused on the inverse problem of ECGI, mine will be more on the forward problem of ECGI, with specifically relating to the passive bidomain and looking at the uncertainty quantification of conductivity in this context. And so just for a little bit of math and context background, the passive bidomain is a representation of the cardiac tissue, specifically during a particular time instant within the activation cycle or within the electrogram. And we are, in this particular study, focusing on the ST40, right in the center of the ST40 or ST segment, which is a clinical marker of myocardial ischemia. And so often the passive bidomain can be used to model myocardial ischemia during this time point, because you'll see a characteristic shift in the ST40, as you can see down here, from baseline when there is a presence of ischemic tissue. And so what the passive bidomain does, if you're not familiar, is we take the tissue, which is canonically represented with the fibers, intracellular space and extracellular space, and we represent it as two overlapping continua, a bidomain, the extracellular space and the intracellular space. We then define parameters of each of these space, being the extracellular potential, phi e, the extracellular conductivities, which is an anisotropic tensor, and then the same thing for intracellular values, potential and conductivity. We then defined a couple more quantities, the bulk conductivity being the sum of the intra and extracellular conductivities, and then the transmembrane potential being the intracellular minus extracellular potentials. And then finally, we use the bidomain equations, specifically the parts that don't have to do with capacitance or changes in charge, to link these two domains together. And what this is essentially saying is the current leaving one domain must enter the other domain. So any current that leaves the intracellular space goes into the extracellular space and vice versa. And so using this representation, we can solve the forward problem where we know transmembrane potentials and we want to solve for the extracellular potentials throughout the heart. This can also be extended to solve for extracellular potentials throughout the torso as well. But what are the parameters that I care about? Well, I told you before that I'm caring specifically about the conductivity values. And so for the purpose of this study, I broke them up into longitudinal, transverse, and then the blood conductivity, which is isotropic. And these are just the conductivity ratios that I've seen used in literature, both by our group and other groups, to describe the conductivity within each of these regions. And so what I wanted to look at was varying these four conductivities here. And in lieu of really a rigorous body of research on what these conductivity values are during ischemia versus during healthy tissue, I just varied the 4 ischemic values during by plus or minus 20% of their kind of standard default value. And this is, an approach that I've kind of talked through with both our uncertainty quantification math people like Akil, and talked through with the research group about, trying to approximate what I think the uncertainty could be in this case. And so after doing so, I did this first in a simulated data set where I generated a sphere of ischemia by setting transmembrane values within this sphere to be at ischemic values and then 0 everywhere else, and set a transition value in between. And so I'm going to be showing you the visualizations of both the forward simulations and the uncertainty quantification output in this simulated data set. And so here baseline is representing the baseline forward solution. So these are the solved extracellular potentials throughout the heart, just using the default values that you might find in literature. And so as you might expect, we see a ST elevation or high potential values at the region of ischemia, and it's flanked by some ST depressions. And then it's mostly 0 throughout the rest of the heart. After that, from my uncertainty quantification, I found the mean solution, which is very remarkably similar to the baseline solution. And then I also found the standard deviation. And as you can see, the majority of variation due to changes in these conductivity values happens mainly within the elevation, and it's not, and then some small extension into the flanking depressions. So once I saw this, I thought, okay, well, I want to know which of the conductivity values are having a predominant effect on this model output, which of the conductivity values are affecting my solution, and which ones maybe are having less of an effect. And so here I'll be showing the total sensitivities for each of the individual parameters. I won't be showing the parameter interactions because there was not very many parameter interactions. As you may remember from before, these sensitivities are normalized to be on a scale from 0 to 1. And so this is the extracellular longitudinal sensitivity. And as you can see, there's kind of high sensitivity right within the region of interest. And then it kind of makes this very confusing looking swirling pattern that if you were to look at the fiber angles, then you would maybe notice some similarity between the directions that the fibers are pointing and the direction that this swirl pattern is pointing. But what I found interesting is that when I looked at the extracellular transverse conductivity sensitivity, it was 0. It was practically 0 everywhere. So the variation that I had imposed on the extracellular transverse conductivity was almost nothing. Then when I looked at the intracellular longitudinal sensitivity, I kind of saw an inverse picture of what I saw in the extracellular. And then the same thing for the transverse conductivity, I again saw almost no variation. So then using these sensitivities and standard deviation metrics that we get out from UncertainSCI, we can actually visualize what would be the standard deviation contribution of each of the different metrics, or each of the different parameters of interest. And so in this case, I'll show the standard deviation contribution of the longitudinal intracellular and extracellular. I'm not showing the transverse because it was 0, because the model wasn't sensitive to them. And so we can see that the extracellular longitudinal mainly contributes to the standard deviation within the ischemic region, and the longitudinal intracellular has more of an effect around the ischemic region, a little bit within and closer to the endocardium. And so then I wanted to extend this into using some not simulated data. So the not simulated data comes from one of our large animal experimental models where we measure myocardial ischemia within the wall of the heart. And so what you're seeing here is the ischemic zone that I have derived from these extracellular measurements. This is taking those extracellular measurements, thresholding them, and then using a rules-based approach to generate transmembrane potentials based on the measured extracellular potentials, which we presented at a previous conference. I then looked at the measured extracellular potentials, compared that to the baseline forward solution, and then the mean forward solution and the standard deviation. And what I saw is that there was the highest standard deviation right here on this highest portion of elevation, which seemed like it may be clinically relevant, that we're getting a large amount of standard deviation in the region that we actually probably care the most about, this elevation that is actually the marker of myocardial ischemia. And so then I went through the same visualization of before of seeing the sensitivities. And as you can expect, they followed the same trend where longitudinal had an effect and transverse did not, both in the intracellular and extracellular domains. The pattern here is much more complex and difficult to parse because of the organic and complex shape of the ischemic regions. But again, when we kind of simplify it down by just looking at the standard deviation contributions, we can see that the extracellular longitudinal seems to have the highest standard deviation contribution, followed by the intracellular longitudinal, and then the two transverse conductivities having almost no effect on the solution. And so what I would learn from this, if I was trying to apply this knowledge going forward, is if I wanted to, say, do a parameter estimation of some sort, or solve an inverse problem, or personalize a model to a certain patient, then I would not necessarily care about trying to tune the transverse conductivities off of their literature values, because clearly tuning those conductivities isn't really going to affect my solution very much. I would, however, care about tuning the longitudinal conductivities, and especially the extracellular longitudinal. And so this kind of helped me understand what parameters are more or less important in this particular simulation. And of course, I only selected 4 of the many parameters that I could have for the passive biodomain. So future studies, we would love to explore that more. And if you want to hear me talk even more at length about this, I will be presenting this research at CNC 2021 in Brno. So thank you.
Thank you, and hope you enjoyed that video showing a couple of applications in the domain of cardiac bioelectricity simulations, where I hope you can see just how useful the UncertainSCI tool set is for exploring questions about the role of parameter sensitivity and the importance of different parameters in the solutions to these types of problems. If you're interested to see more, please visit our website, read our documentation, and reach out to us if you have questions that we can help you with to get you up and running with UncertainSCI.